Well, hello and welcome to another one of my thoughts videos. In this one I'm going to try and get the mitochondria sorted out as far as I think it works rather than what the mainstream thinks it is. So let's start with photosynthesis, and I'll do some drawings. Now, starting with photosynthesis, you have your chlorophyll in a plant's leaf, and the sun comes in, hits that, and it gets the water, which is an O and two protiums there. Now, in the little, in the chlorophyll of the plant, more pointy leaf, the manganese group, it takes that light, makes it like a big magnet, and sucks electrons off, at least 3, so that liberates the protiums and the oxygen, and the oxygen gets used up in other processes if it doesn't get thrown out into the ambient as well. Now, those two protiums, hydrogens, they get joined to the ADP. The P is a phosphorus. We have that in our bodies, but it's used in another system to do with genes and stuff, and a whole bunch of other things. And so that becomes a P and an H. So you get 2 of those, NADPH. So those 2 there then transport the proteins. Now, as I've said in other videos, I think the 2 protiums are the currency, not the electrons. The electrons are incidental and they are useful, and those are passed on to other substances, or what do we call them, proteins and chemicals and things and atoms, and they get passed along into the mitochondria as well and get used in the process. But I think the protium is what is most important. So that's how it is absorbed there. Now, to store those protium ions, the hydrogens, it grabs 1, 2, 3, 6 carbon dioxides, and but it doesn't want the oxides, it only wants the 1 oxygen. So it has 1 oxygen here. The next one we have a carbon monoxide, and then it puts it back to front there, and then on that side, and then a funny thing happens here. It's going to now put those protiums, and it's going to store a whole heap of them. Only 6 oxygens and 6 carbon dioxides, and 6 oxygens are thrown out. So from the carbon dioxide now has only got 1. Then it's going to add a protium there, a protium on either side of all of those, sort of like a fish skeleton. But it's going to stick 2 on the bottom one there, and an extra one, so it's got 3 on that. So because it's got 1 up here, it's got an extra one down there and stuff. But that's your glucose molecule. Fructose is slightly different with 1 more up there. That's complete, but it's got the same number of protiums and carbons and oxygens. So that's the glucose molecule that then string together with another and another and another until it's a great big long chain, and it is stored as the sugars that we know. Now, that's the transport of what I think the important part. They're all joined obviously by electrons, but the important part for me is considered that the carbon and the oxygens from the carbon dioxide to now carbon monoxides is to transport the protiums. Now, when the plant or an animal wants to use that glucose, it gets it into the system, and then in the mitochondria you have an outer membrane, and then you have an inner membrane that does this, and you're talking the difference between this gap here of only being 8 nanometers, something like nanometers, it's very small. Now in that membrane here is where the interesting things happen, where the respiration happens. Outside here, outside the mitochondria, the glucose is split into two pyruvates and then brought into the mitochondria. Now that splitting of the glucose into 2 pyruvates is an anaerobic situation, and it loses 2 of the H's into an NAD. It adds those and it loses 2 of the 12 proteins to the that process of breaking into the pyruvates. Now the pyruvates come in, and in the inner membrane there are proteins in that membrane. Now, it takes it in there, and through the Krebs cycle, or the citric cycle, sometimes they call it, and I think it might have another name, but that goes through about 10 steps to dismantle that that glucose molecule, wherever I have it. Now the results are it needs oxygen in those proteins in the membrane, because those 6 carbons, which are now split into 3 and 3, it needs to have extra oxygens to take the carbon and the oxygen in each chain, and there's 3 of those, and it has to add oxygens to give you 2 oxygens, so it can give you the carbon dioxide. So you're going to get 3 carbon dioxides out of it to get rid of the carbon and the oxygens, and then you're going to have more of these NADs made into H's. There's going to be 3 of those, and that accounts for 3 protiums, and you're going to have one, and FAD, it makes 1 of those, and that is 1 of those, and then you'll also get 1 ATP with the extra electrons that were spare, so that helps there. So it's now dismantled. Those carbon dioxides are thrown away, and as I say, I believe that the hydrogen, the protium ionized protiums are what's most important. You've got 4 here plus the other 2. You've got a total of 10 of those that you're going to get. But I noticed here, doing my math, your FADH, that's a double one, so you've got 2 H's on the FADH, 2 protiums, and you've got 3 there. So there's 3, 4, 5, so that's not 2, that's the 5 there. Double that, and you've got 10. And of course, plus the other 2 from the other process of splitting the molecule into 2, and there's your 12 protiums. Now, in the membrane, continuing on here, current thinking says that in these proteins, as they're broken down and passed from hither and thither, they say that the protiums, the hydrogen, ionized hydrogen protium ion, is passed out into the outer membrane, or into this gap, to then go around to this motor, which is here, which is the ATP motor, and supposedly it goes past, making this spin, and then the ADP, that's adenine diphosphate, gets another phosphate group which actually has 4 oxygens to it, and puts another one on there using the electrons, which they say passed inwards with the protiums outwards. I totally disagree with that. I believe that the protiums are sent inwards, the protiums inwards, and the electrons outwards. The electrons, or put them like that. Now, why is that so, and what's my reasoning with that? It's to do with motors. The people who designed this did not understand electricity and how motors work. Now, it's all to do with electricity, and as I've said in another video, the fourth state of water contributes to this, and the protium is the protium ion is the other half to the story. Now, on the outside of that mitochondria, a layer of the fourth state of water up to 200 nanometers deep is going to coat it with a negatively charged water structure, crystalline structure. It will fill the entire structure within the between the two membranes, also because that's only 8 nanometers, and so it's going to fill that up with crystalline water, and it's also going to start filling up the crystalline water in there, so it's going to be all negatively charged. Now, outside of that, it's neutral, because water is basically neutral, but when it turns into this layer of the fourth state of water, crystalline water, it takes on it gets a negative charge because it loses some hydrogens and things like that, or for some reason, which I won't go into detail right now, but it becomes negative, and so this whole structure is negative. Now, when you get to your situation with the inner membrane with the Krebs cycle, it is more logical that the protiums go inwards rather than outwards. Now, they only have 4 ways of testing whether they are going in or out, and I've looked at those, and there's spectroscopy, acidity, and 2 others, and all 4 of them are open to debate and open to error. Now, from an electrical point of view, they should go inwards, because they have lost their electron. They are now positively charged. Because they're like a proton, but they're not a proton. They are a protium, that is a hydrogen, a protium, an ionized one, which means it's lost its negative electron, so it itself is positive, so it has no electron to balance it, so it is now positive. Now, remember, this is going to be negative, and this is going to be negative also by nature. And as I said in other videos, there's electrons floating around all over the place in great abundance. When you're setting up a component, and an electronic component, you need to give a reason to control and make electrons flow to where you want them to flow. In this case, if you have electrons on the outside due to them being passed out of the Krebs cycle, and as well as the negativity of the crystalline structured water anyway, you are going to have an abundance of negatives, that is electrons. If you pump your protiums into the center, you're now going to give a more positive charge to it, so that by the time you get your ATP synthase, that is a motor. Now, it has a stator thingy on it like this, which goes down. I have to look up. I'll see if I can get you a picture of that. But it has a head and it has a body where the ATP is situated. Now, because you have a negative, lots of electrons with the fourth state of water and the electrons coming from the Krebs cycle, you've got a positive side here. It's going to make them want to flow. The ATP synthase gives a path for the electrons to then flow down and into balance up the charge of the inner side of the membrane where the protiums are. Now, as those electrons go passing here, what these biologists and don't know is that on this part here, there's a part that is aquaphobic, and so therefore acts as a bearing and lets it spin. As those go past, they create a magnetic field. As we've said in other videos, the electrons don't have energy. When they flow or move, they create an electromagnetic field around where they're flowing, and that creates a mirror electromagnetic field in the ATP synthase, the rotor part of it, and it repels those, because this is magnetized. This is magnetized, or there's a magnetic field and a magnetic field, and because they're the same mirrored magnetic field, they repel, and it makes it spin. So now that that's spinning, and now as they go past, they're in great abundance for the mechanical part down below to take the ATP and use those electrons that are flowing past to then take some of those from the flow as it spins and paste the extra phosphorus group to the other two. So it's more logical. The electrons go out, flow down through here to then try and balance up the charge, and in doing so creates a magnetic field which makes the motor go, and then the flow past the bottom of it also has abundant electrons to then paste the other P on, and then those go further. Then there is a four-step step to it's in here. It's a four-step process to break the hydrogens off. The first stage and second stage is to break the hydrogen off. It gets off the NAD and the FADs, and then they get passed to another, and they get passed into here, and then eventually with oxygen and the abundant electrons in the oxygen, and another one here, the electrons that are coming through, there's an enzyme that then joins them back together to the water and turns those protiums and the electrons coming around here back into water. Now, to me, that is a way more logical thing. I hope it wasn't too waffly. I hope you can get that. Go back through the video and check it again. But it's a motor. The electrons are passed out with the help of the fourth state of water. Protium's in to give you the positivity, so that it'll cause an electron flow past the ATP synthase, making it spin with the magnetic fields it sets up, and giving the electrons to paste the P on, and the extra P, and then that is your adenine diphosphate and triphosphate. Now that is where you are now saving an electron here by joining the extra phosphorus on, but your hydrogens, your protiums have done their job and turned back into water. So you can see that the important part that I'm seeing is that the protium, the H from the water molecule, is what's most important. Electrons are abundant and fine, but they need to be transported around to make components for the electrons to be controlled. Now the protiums are stored in that glucose molecule, or fructose, and that is what gets stored so that plants or animals can use it at a later date, so that it can be dismantled and used to form the component which is going to now make electrons flow to get that final useful product, to have lots of individual electrons trapped in that ATP here, which then can be used to generate electrical fields in other situations.